Yeah. So, hello everyone. Good evening. And so today we are in the week 5 of the live session where we are trying to, where we will be seeing that what is the different applications, and specifically also we will look at the scanner. So we have looked at the different microarray applications and the different microarray techniques, but we have also looked. The scanner is a very important thing for scanning and for getting particular the microarrays, but majorly what it is required is that we are trying to understand that what particular uh, scanner is, how the scanning works, what are the different parameters that needs to be understand. Then finally also what are the different applications like the auto antibodies for different cancer research, and also then finally we are going into we'll be seeing the reverse phase protein arrays of and what is the major difference between the forward phase arrays and the reverse phase protein arrays. So now moving on, before moving on, I'd like to show you the video once again if you have. Yeah, so this is the thing, so how the scanner looks like and how it is coming. So now just for you again to visualize it, so that so these are the different uh, icons in the lecture you must have seen, but this is how typical software, the GenPix Pro of the particular machine, which it is being referred here, and which of which we will talk for the understanding that how particular scanning is done of a microarray. But just look at the interface. I am sure you have gone through the lecture and have seen it, but again, see it directly from the lab that how this particular thing looks like. So this is a particular grid. So the gal file which is being told that how that particular alignment is done for the particular regions and the for particular spot to extract out the spot intensities. So if you just remember, just look at the wavelengths, different wavelengths, 635, 532, through which you can analyze and you can try to uh, the different laser which the excitement happens, and to get the particular extract the intensities out of that particular protein for different dyes through which it is labeled. And these are the different tools for the zooming in, zooming out, and the tools that have been mentioned. So just look into it once again. So in the previous week also had shown you the particular video again. This time with the context, you can see that the gal file for the alignment. So also you have seen in the lecture that you can do the auto alignment, yeah, and it will get aligned properly and automatically. So just focus on this particular. Here you can see that there's an intensity which is showing that particular spot has got certain protein labeled, and you are seeing that certain fluorescence is being coming out, which is marking that here that certain interactions has happened and what, but here there is no. But still we cannot know what is the particular intensity level, and we need to do the extract it out by doing the background correction and normalization, and then only we can get. So in that context, how to understand that what is the particular extraction level and what is the particular gain for this, and how the intensities doesn't get the intensities don't uh, get exhausted out in a way, and the particular intensities doesn't turn out to be very much that some lower level noises are missed out, and the higher intensity noises are have been very much saturated, which will give us the false result. So that adjustment needs to be done, and that's how we can get into the optimal intensity level through which our optimal amplification in that matter for the electrode voltage we can get in that results. Now we can see that these files are layered. These are the at the different channel, at the different extraction level. So now you can see, view the blocks. Correct. So you will need to do the alignment properly, and you have the information and the annotation. So these are, if you remember from the lecture, which you will again see that there are the different features, regular or irregular features, which we can get at. And also in the background, if you see, so these particular regions are something you can see that there's no protein, but during the washing and other steps, this kind of state and this kind of uh, fluorescence you get. Right? So different features and the different shapes and sizes, you can try to build into that. In the different channels you are taking. So the alignment here is the key to extract out the background information. Okay, so now you can see that the result. So again, just so before, since you have all have gone through the lecture and have found out that the histogram and all these things, scatter plot and everything, so you can see in the real scenario and in the real machine also this kind of options are there, and you can look at the histogram and all to understand what is the exact level, and so that you can adjust your gains and adjust your parameters accordingly while the scanning, which is very much important for this kind of things. And see, you can see the extract. So these are the annotated informations, that what particular region, what particular spot corresponds to, and what is the intensity, foreground and the background levels at which channel, right? You can see. So this is the Excel file which you can export it out. Okay, so I'll just run it once again for you all to visualize the whole procedure for analyzing and getting. Okay, I'm just running the process you out again to get you familiarized with the tools, different channels, and how this analysis is done. So this is from the real time in the lab setup, how it is done, and whatever we have gone through the lecture is available here. So you can see, adjust and align those to extract out the features and all the annotated information that provided. So this is quite important in a way to understand of what context we would be talking about, so that you can get a proper idea of what we are saying and why this is important. Apart from doing the experiment very meticulously with all the reagents to be in proper place with proper sample preparation and the proteins, this also plays a huge role if when we are doing using high throughput arrays for understanding of the any particular thing. So again, this is your particular thing that it is also a kind of a that what we showed is a peptide arrays, which is for your epitope mapping, which you need to understand and which you can have it. So now, what is the design of this kind of scanner? So we have seen so how this scanner looks like. I'll just go to some extent before going into the front. So this is the kind of a scanner which you will place and insert the chips into it, and this is the video where you can see that how particular things are getting up, and you are aligning the spots and extracting the features out. Now, what is the major major principle? How this scanner? What are the parameters? So now the thing is that there is, of course, you can understand that there are a lot of physics and electronics associated component associated with it. So you have the scanner, you need to scan it out. But if you are doing the experiment and if you are uh, as a biologist, you are have designed one experiment and want to get an idea that how a particular system and particular or this thing works, and particular uh, scanner particular experiment, and if you are after you have done the all the collected the samples and have done all the proteins and chips which are microchips which are also very costly in nature, and if you are bringing and you are doing the experiment, so you need to have a proper understanding that how this particular scanner works, how this particular adjustment needs to be done for that particular scanner, and then you need to adjust your parameters. And once you have adjusted your particular parameters, then only what you can do is that you can get this particular right information out of this particular thing, and then can extract out and get it the result done. So now we will look for the scanner design. So the hardware, that what it happens is that, so this is a kind of a non-confocal design. So what we are setting up that the micro scanners design is non-confocal, whereas in the confocal with the layers the images are created, but here it is non-confocal design. Why non-confocal? The thing is that the background and the spot signal is in the same plane of the focus, that is in the same focus the background and the spot signals are also placed. Second thing is the inverted optics. So these are the two major thing that is required, or the two major hardware principle on which the micro scanners works, like the GenePix scanner works, which inverted optics is there due to the measuring the deformities of the glass slides, and to have the good signal to noise ratio. And then the slide is always printed on the inverted uh, supported on the printed slide, and excitation on the same side as the measurement of the fluorescence signal. So whichever side the fluorescence signal is measured, the excitation also happens on the same side. So you can understand that this is a scanner. You have the printed slide. Okay, so slide is supported to the printed side, and you have the excitation source. So it will measure the fluorescence signal. So the laser light will come and it will measure the intensity out. So this is known as your inverted optics. So these are the two particular aspect of this kind of hardware design. One goes to the non-confocal, and one is the inverted optics. So why these two particular reason is there, or particular thing is there, because it will help you to get the major thing or major thing that we want to achieve is that we have to need to have a good signal, and other than the noise, and then it should have, we should get a good intensity and this thing. So that's why this is the kind of a hardware design that is happening both so majorly the non-confocal and the inverted optics. So what machine I had showed, so this when we place on the chip in this particular machine, and when the scanner is happening, so it is belonging to this thing. One is the non-confocal and then the inverted optics, that is the excitation is happening and it is taking the uh, signal out. Okay, so inverted optics and the non-confocal design. Now, next is the major very important component of this microarrays. The scanner design is, that is known as the multi photo multiplier tube, or the PMT. So what it is that? So you can see that it is presence of different anodes and cathodes and with different resistors. So what it happens? So you can understand through the name that it is multiplier, right? So it is a multiplier. Photo means coming from the term of the photons. So what it does is that whatever photon comes, photon means the light wave particles. So it amplifies the signal. So why it amplifies the signal, or amplifying the signals is required for the detector to detect it, to extract out the features. So majorly, if the intensity of particular protein is very, the expression level of that particular protein in any condition, be it disease condition or be it normal condition, is quite less, then what will happen? So then that happens is that the signal won't be reaching to the detector, or won't be getting, we wouldn't be getting any such as such different things, as such different signals, and our signal to noise ratio would be very weak and we won't be able to detect. So the major thing is to amplify the signals. So now the detectors multiply the current produced by incident light. So the major task or major goal of this photomultiplier tube is to have that the detectors multiply the current produced by incident light as much as 100 million times, or 10 to the power 8 times, by 100 million times, or the 10 to the power 8 times. So the goal is to amplify the signal from weak intensities to observe and measure, but also not saturating from the strong signal. Now, whose expression is quite strong and giving you a good intensity. So, for example, I'll just refer to the video once again, because I'll just, yeah. So here you can see these signals are quite strong, right? Now, if you are gaining and it is becoming your PMT gain, you are increasing, amplifying it to such extent that what it happens is then it will get saturated, and that won't be the exact situation we want. We want that get the amplification from the weak signals, but not saturating it from our strong signals. Where that's where we need to keep in that's where we need to keep in mind the things that we cannot afford to lose that track and to lose that part, correct? So we cannot lose that track or lose that part, that we are just decreasing or we are increasing, and we are not able to get and manage that part, right? So this is quite important, because that's why getting a good PMT gain is quite crucial and important, specifically when we are doing this kind of particular experiment, right? So this is the goal, is to simple goal is to amplify the signal from weak intensities to observe and measure but also not saturating from strong signals. So I repeat again, not saturating from the strong signals. So we cannot saturate it from the strong signals. What we can just do is that we can amplify the signal from the big intensities. So that's why PMT gain, photomultiplier tube gain is very much important. Okay. So now one important aspect of whenever we are looking at to it is the signal to noise ratio, right? So what is signal to noise ratio and what does we mean? Correct? So here we mean that, so signal to noise ratio's basic definition is to measure how well true signal can be referred from the noise of the system. Each and every system has some baseline noises whenever we do and whenever we take. So these are all noises that gets generated due to different electrons and photons randomly jumping and changing their positions. So we need to measure that how well the true signal can be resolved apart from the noise of the system. And then next point, which is very much crucial or vital, is that the brighter feature only does not correspond to having a greater SN ratio. Might be the background of that feature is quite high, which is leading to a very high signal, but that doesn't correspond to have a greater signal to noise ratio. So always it depends on the background and the background level of the noise. So the ratio, the signal to noise ratio, it is the signal power and the noise power, which corresponds to the actual the ratio between the signal to noise. So this is how the things are. So this is a signal, this is a noise. So this all these which are at the baseline level and noise, this is a signal. Now, for instance, if particular signal we get this much or a little bit above the noise, a little bit above a peak we get. So whether that is a signal or whether that is a noise, so that is a threshold we need to give to the instrument, and we need to provide that to let us understand which is a signal, which is a noise, which is the cutoff we should use, we should not use. So that's the ratio and that's the threshold we need to provide to the system for us to get the extract of the basic feature and basic out of it. So this is the simple formula between the SNR, that is the signal to noise ratio, ratio between your signal power and the noise power. So the signal to noise ratio is the P signal divided by P noise, which is the uh, amplitude of the peak uh, at that particular thing, and this is the formula at the decibel level. So this is how the signal to noise ratio is done. So now if I refer to again that, you can see that there are some brighter features, some lesser features. So after you have done the photomultiplier tubes and you have engaged it with the different particular uh, gain and amplified it, now you need to have a good signal to noise ratio, so that to get it. Okay, so then only you will be able to extract out the exact information without having any instrumental or technical very technical technical noises, rather than the biological information and the biological signal. So to have an understanding that what signal to noise ratio and what cutoff or what threshold should we provide. Now there is a concept in this kind of instrument is the scan line averaging. Now the thing is that if you scan it one time and you get certain thing, and whenever you are doing it uh, for 3 or 4 times, definitely what it happens is that we can get an idea, or we can get an information, and we can do an averaging, which reduces the background noise, which reduces all the technical glitches, all the technical problems that happens during one time, are will be getting to your changed whenever you are doing it for 2-3 times. So this reduces the background noise. So the main idea and the main goal here is to reduce your background noise at. Second. One second, just. Yeah, so to reduce the background noise and to increase the number of accepted spots, so that's what we are showing, that this number of spots, if there is certain difficulties having a high background and low signal for from the apart from the other rest, then we can lost these spots which can be useful, which contain and which might contain an useful information from the biological point of view, but might get lost because of this technical problems and the glitches. So that's why by averaging scanning the average, but also the one thing that we need to keep in mind, that we cannot have a huge amount of time exposed these particular proteins to the fluorescence light, which will be again detrimental for the thing and can be lead to other technical issues. Just give me a minute. Yeah, sorry. Extremely sorry, this is some issues. Yeah, so where we were, so I was just, yeah, telling that. So yeah, so if the extraction doesn't happen, and also there are different spots like this one which you were seeing at the edges of the different control spots, which will be showing the intensity, and it's also a measure that whether our PMT gain and adjustment gains and everything are fine or not. Otherwise, it will lead to disruption, and it will lead to a variety change in the uh, variety level, and also not help us to do the background correction and normalization in a correct or proper manner. So that's where it is becomes very much important and crucial. Yeah, so it's good. Yeah. So what is the important advantages? One is the reducing the background noise. Yeah. So 1 second. Yeah. So one is to have a. Yeah. Okay, so what we are saying that it reduces the background noise and we can increase the number of accepted spots, increased overall is SNR. So signal to noise ratio will be better, which we can see. And finally, balance of the added scan time, a small gain in the detection limit, and increases scan time as multiple number of scan times to detect. Again, I said there might be certain issues with the increased number of time through which fluorescence intensity gets particular fluorescence intensities are getting uh, your particular exposed to this fluorescence. So the thing is that, so if you are getting signal and these are the noise, signal and the noise, so if you just do it, might get it, and will leverage will help you to have a better SNR and the added scan time and the detection limits. This needs to have a balance in between them. Okay, so this balance is something required. And scan time has multiple number of times to scan, but overall it is better and helpful for us. So this is the microarray system of what we are referring to and what we are showing, of which the chips, like the chips which contains different spots, which gets entered into here through the inverted optics. Getting the intensity out. Getting the intensity out. So this chip, what is the major advantage of using this scanner? That is GenPix Pro. So this is the real-time PMT adjustment. So that is what we know as auto multiplier. The PMT gain adjustment. So what it is known as? It is known as the PMT gain adjustment, right? So what is the goal of the PMT adjustment? Amplify the weak signal without saturating without saturating the strong signals. Very, very important. So this can happen in this kind of scanner in real time, of which we are showing you the video, and this one which can happen in real time. Auto PMT adjustment. So automatically, if you ask the instrument to, it will judge and it will do it so that it doesn't affect our signals. And then the line averaging. So this helps us to get. So what does line averaging helps? Again, I'll just repeat for you all. So in the line averaging, so it increases your overall uh, that particular uh, spots and SNR. So it helps us to loss of the particular spots and particular features, otherwise which will be missing out if it is biologically relevant also. Okay, so this is one thing. So now this data acquisition, of which I have shown, I'll run it once again for you all to go through it. Is go through it very carefully, the complete entire process that will lead you to the understanding of the data acquisition, and then we are coming up that what is the feature data. So again, for you all, look at carefully what it is. Okay, see the complete data acquisition procedure. So just try to understand that what, after you extracting out the information in the Excel is fine, but what is actually it is. The images. I'll run it once again for you all to check, and then we will look into the different features. I'll just look at every step very meticulously what it is happening. I'll run it once again for you all. Run the video once again to see you how the extraction, because this is what now we will be mostly focusing on. One was the scanner characteristic. Now after the data acquisition, what are we doing? It's very, very important for this kind of experiments. Okay, so I'll just play it once more for you all to understand it very thoroughly. Right? Yeah. Look at the entire procedure once again. What with what we would be playing upon to get this kind of particular things. So this is a real thing, you are getting the experience directly from the lab, which will help you to get an understanding much deeper way. Okay, so I'll run it once again since you are for you all. So, because again I'm saying, please look at each and every step very carefully. This is an extremely important step, and after this only we'll be mostly focusing on different analysis and different extraction procedure. So please go through it once. I'll show it once more for you all. Okay, so yeah, so this is once more. Just look at it. It's a very crucial steps. Yeah, features, the channels, the way these blocks are being aligned, finally extracting out the features. So this all you can do. So we'll come into the modes as well, that how to understand when to do auto PMT adjustment gain, real time PMT gain adjustment. What are the steps where we need to do it? So all things are very important and crucial in this aspect. Yeah. Okay. So now we will look at the data acquisition. So what we are saying that it's multi-image that the multi-image or the single image in TIFF or JPEG format and the image alignment. So why I stressed out on the part that please watch the video, it's the alignment. So once you get that block, alignment is the key. If we the square or in the irregular features, if we are not aligning and have a manual intervention, manual intervention, we would be extracting different information. So we would be extracting the exact information, rather we would be having different information that we would be extracting out, and that would lead us to the different properties. The feature, some we can have more background, we can remove that extracting out the feature. So that's why the alignment and the block and feature alignment is very crucial. So that's why I told repeatedly to look at the video very carefully and to observe each and every step, because that is our most crucial. Once you have done the experiment after such a lot of hard work, all the costly chips, you have done the experiment, precious samples, clinical samples which are difficult to obtain, and if a single mistake in during the data acquisition and the scanning can ruin your complete experiment, and it will be a huge detrimental and setup for your particular project or particular thing, or trying to observe certain objectives. So that's why I repeatedly said that each and every step in that video what we have shown is quite important for you to get an understanding and get a feeling of exact lab setup, which helps us to understand. So now the scanner data acquisition, the background subtraction can be local, global, negative controls and positive controls. So what it is meaning that at particular spot, no intensity should come up. Should come up. Okay. Now this is a negative control, but still there will be some background intensities due to the bleaching or due to different washing steps. So you have placed the primary antibody, secondary antibody, wash didn't happen very well above also, though you have so that's why in the previous week it was important to understand the steps, that how the required. You need to have a different steps of the washing, the incubation, washing in the using different rockers, which we had shown very meticulously that how it is to be done. Still, what happens after you have done all the steps very meticulously, still there will be certain percentage of that the washing doesn't happen, secondary antibody remained, there's some kind of bleaching happens, there kind of some kind of intensity. So the spots where no intensity should be observed, so there is certain background comes, and once the background comes there is certain intensity. So we So this is the background noise. So whatever intensity we have, we can directly minus from this background. So this will give us the background subtracted So these all gave us background subtracted intensity. So these all what it gave is the background subtracted intensity, right? So this gives us the background subtracted intensity. So that can use in local, global, negative controls, etc., etc. So all this will lead us to the background subtraction, ratio calculation of the different wavelengths and the different channels. Now the normalization of the images, again, that's why I stressed out to look at each and every step of the data acquisition that we were showing directly from the lab to have an idea that what particular goes into. So the normalization of the images and the numerical data to understand and showcase. So the extracting annotation information from their images, you can see that the red and the green different channels. Now we want to have the numerical data for you to further analyze, further finding out certain features, certain markers, candidate proteins, which are having a different expression and different things in your particular body. So this is the major important chunk and important thing. Then display the background pixels on the image and the histogram or scatter plot for any analysis measurements, which again I had spotted onto it. Let's check that each for the tube contains the histogram and the scatter plot. I'll again play because I am sure that you have missed some out of this, but now I request again to look carefully on the context that we are talking about. Okay. I'll just play for you once more. Look it carefully. Now look at whatever context that we have talked about. Look it carefully each and every step. Alignment is important. What this scanners gives us the features. Why it is so costly and helps us in getting. See each and every step we need to have a manual intervention. That's why I mentioned. Okay, just now check. Look it carefully on the context of which we have talked about. So from these images, we need to, after doing the background correction and those intensities, we need to extract out the numerical data for further analysis that we are talking about. Okay, so this is quite important. Now I think you have understood on the context of it what we are talking about. So now what are the scan modes that are there? So one mode which you can say is that the preview scan. What is preview scan? So first you need to understand if we are doing, if you want to have certain PMT gain and adjust it, whether it is happening fine or not. So that's why we need to have a preview of whatever we are scanning. So scans, preview scan mode works on what, how does it work? So it is, sorry, just give me a minute. Yeah, so just give me a minute. Yeah, sorry, extremely. So scans all selected channels at 40 micromolar resolution. So what is the resolution and what is the, so that you can differentiate between the two small spots with the small diameters, and scans entire slide surface. So this is specific to one model. Okay, so then is the preview scan mode, data scan. It scans selected region at the selected resolution with selected lines to average. Concept of scanning average. Scans all the selected channels, different channels. Sorry. Then is the single wavelength scan, data scan to only one of the selected channels and reduces the exposure to the lesser light. So what are the scanning modes that we have talked about till now? One is the preview scan. Second is data scan. Third is single wavelength scan. And fourth is the preview scan and then data scan, which is rarely used. One of the methods is rarely used, and it is usually when scanning lots of the same slide type on the single slide scanners. So these are the four modes. One is your preview scan, have the data scan, single wavelength scan, and preview scan, and then data scan. So which is rarely used because it will lead to a lot of laser exposure on the samples, which is not good. And also when we are using the scanning lots of the same slide type on the single slide scanners. And others are the preview scan, then the data scan, and then the single wavelength scan, that each particular channel which we can do the scanning. And it also reduces the exposure to laser light, which is good in a way for the samples and those things. Okay. So now, so what is the image capture that can happen? So what are the features of this kind of instruments of which we are showing and talking about? That it can simultaneously acquisition of two wavelengths. I think now you should be clear and have seen and observed very meticulously. If not, again I will show it, because if you are familiar with each of these features and you are getting this one-hand experience of it from the actual lab setup, you will be more confident of using or handling this kind of instruments. Then is adjusting the PMT gain. So this is by the manual intervention or auto or in a real time manner. Again, PMT gain is very crucial and very important, and it's also been emphasized, needs to be emphasized, because it will give us the intensities from the weak observations, weak signals to amplify to such an extent that it can be detected and can be measured. At the same, it will be that it should not saturate our own, that it should not saturate our own from the strong signals. Adjustable laser power, right? Adjustable laser power, 5 to 100 micromolar resolutions, adjustable focus, line averaging, and quantitative measure of the channel intensity balance to look for the current PMT gain. So this is very, very important. I am again showing you just to for you to observe it in a much meticulous way, and please try to understand with the context of which we are talking about. That will help you to understand the system much better way. Okay, please look at all these features once again of which context. So that's why I am repeatedly talking about the system, talking about the different parameters, talking about the different terminologies, and showing you the real how it is done on a real scale from a real lab setup, so that you can understand that what we are talking about, what is the actual context. You can see the different channels, different wavelengths in the preview mode, scan mode, and the single wavelength scan mode. So what it will reduce the exposure to the lesser time. So each and everything of what we are talking about is taken care of whenever this kind of experiment is done in a real lab setup. So these are giving you an on one hand exposure to for you to understand it in a much better way. Now the background correction. So now try to understand which of the following is a positive control. Now you should start questioning yourself, how which of these blocks, which are the positive controls, from where to subtract the background. There's some negative spots, there are positive spots. You can now start thinking, and from this context of the real experiments, so then only you can understand that whatever context we have talked about, and we are talking about how important or crucial it is when we are talking about the real experiments. right? So this is all about the scanner structure and how it is done. Please let me know if you have any queries or if you have any questions regarding it. So I tried to give you the basic design architecture of this kind of scanner, how it is done on a real lab setup, and finally we can understand that how it works. So now we will be moving on. Please let me know if you have any queries. You can uh, ask. Now, if we go into the application, it's fine. We know the technology. We know that the microarrays are there. We know in the first week only we have seen different experiments. Now, what is the contextual application of these technologies? So we are either buying this kind of UPROT chips, which are very costly from different vendors, or we are printing our proteins. We are doing NAPA, which have been explained by Professor Laber, Professor Joshua, to a great extent, and then what we are trying to find out and try to achieve it from it. But finally, what we are trying to find out, like to understand the disease biology. So our last goal of this kind of experiments using this kind of technologies is to ultimately lead us to, in a better way, understand what is the disease biology, to have some candidate markers through which we can diagnose the patients for different diagnostic aspects and different prognostic aspects. So this is what finally we need to do and we need to understand, right? So what should be the phases? So one is the discovery phase, one is the validation phase. So if we are trying to use this kind of technologies in understanding different markers, so then we need to go through those two phases, which is the discovery phase and the validation phase. And Professor Joshua has really given a extremely important techniques, important considerations in the clinical proteomics or clinical biology or clinical disease biology whenever we are working upon, that we cannot claim anything to be seriously a marker or something that we cannot be taken to the clinics until and unless we have gone through thorough discovery based cohort with a greater number of samples, followed by taking validating it on a different cohort of samples, because we cannot uh, take it lightly, because that can be a sample specific thing, hospital specific thing, a tube specific thing. I don't know if you remember, as given an excellent example that for a particular disease, that one scientist found out that certain protein is getting very much dysregulated and has claimed big things that it is a marker and can be used in clinics. But after some time, it found out that it was the tube specific and not the nothing to do with the disease and those things. So it is again very crucial for us to understand that whether what cohort of samples we are doing, what cohort of samples we should be validating, what are the techniques we are taking for discovery phase, what techniques we are taking for validating. So if from the biospecimen, if we are taking tissue, serum, CSF, these are the different biospecimens. We are extracting of the proteins. So one proteomics wave can be MSMS, LCMSMS, where we can use level-free quantification, where we can use level-based quantification techniques, which we have talked in detail in the week one. If you have any questions or anything, if you have forgot, you can ask me directly, or you can revisit the week one's lecture recording, which is already available, and uh, uh, you can go through that again. And then the microarray, which is again using a high throughput. So both the things are high throughput and are very much useful for the robust understanding of this particular thing. Second, in the validation phase, there can be Western blot, can be ELISA, can be targeted proteomics. Again, in the week 1, we have discussed in detail about the targeted proteomics, the MRM, multiple reaction monitoring or selected reaction monitoring or parallel reaction monitoring are the emerging methods which have revolutionized the whole field of validation, where we can validate in a high throughput manner, where we can validate the peptides, where we can validate small molecules and can be used for uh, a greater number of molecules which we can monitor and can also be testing out in the clinics for the mass spec based things. And for the ELISA, the traditional ELISA, the conventional methods, which includes antibody-based methods for different applications and can be used for validating in the your different cohorts. Now, these are the some of the case studies which we would be discussing upon, that how particular things or particular features can be used for particular experiments. But just give me a minute. Okay, so now how to get those particular signatures out of it. So this is a study from the microarrays point of view for the autoantibody profiling of the glioma serum samples to identify biomarkers using human proteome arrays. So human proteome arrays are those chips, UPROT chips, which are used for which contains the 20,000 proteins and the serum samples is flown into it. We get those particular proteins out of it and we get the antibodies. So what are the experimental workflow? Let us go through it. So again, so you can understand that whatever we have discussed, so what we have done is that, so what uh, just give me a minute. Okay, so what it is done is that we are getting that particular uh, microarray. So whatever experiment we have shown you, that how to do the meticulous experiments with blocking, incubation, and all those, and the data acquisition. Now, how you can get benefit in the real research or the clinical perspective. We have this blocking, incubation with the patient serums. All those steps have been discussed in detail in the previous week. Incubation with the side-eye, conjugated with anti-human IgG scanning, and the microdata analysis. The things that is written so simple way, you have seen that what uh, 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 are the things that goes into a place and goes into the mind, that how you can need to have those particular things understanding, and then finally we can get. Then what we have done is the quality of the proteins wanted in the microarray. So all the proteins are placed in duplicates. Once they are placed in the duplicates, So what is done is that the same protein, now same sample has the same amount of antibodies, so should get bind to that. So what is the reproducibility of that? So it should have a huge greater correlation. So that's why intra-chip reproducibility is spot 1, spot 2. Now different slides, same sample, different slides. Intra-chip reproducibility, slide 1, slide 2. So this is to be having a huge good correlation. Then only we can say that reproducible, there is no technical artifact, no experiment error, and whatever we are getting is absolutely correct, and we can measure those and to find out the biological differences out of it. Otherwise, it contains no relevance. Okay. Then intra-chip reproducibility, inter-chip reproducibility of the data across all slides pertaining to each group of the samples. What is the distribution of the data? Unnormalized data versus the normalized data. So what are the four things? One is the experimental procedure, then quality of the proteins spotted on the microarray, showing the in-signal intensity with increasing the concentration of the purified GST protein spotted. Now there has been some control. So here you can see this is the GST proteins, right? So what it is doing, increased signal. So the GST protein is spotted of different intensities. Higher intensity should have the higher, whatever that we have learned, that higher protein expression. So that's what we are trying to measure. If certain proteins are getting the particular higher intensities, then definitely uh, uh, those we should be getting it in a higher zone, higher level zone. That's what we are doing, right? So if we have different concentration of the proteins and we are trying to measure their intensity level, so are we getting that particular thing? So if we have higher concentration of the GST proteins spotted on that particular arrays, definitely their gradient should be increasing, and we are seeing the same trend. So this shows that it is correct. The slide is correct, has been experiment has been done correct. So then we can get, when we see that particular spot is showing a higher intensity or higher concentration for a different protein, then we can say that yes, definitely that is biological. That protein is showing certain markers, certain autoantibodies are there, which is not false. Then the intra-chip reproducibility, inter-chip, and the distribution of the data. So now what we are showing, the differentially expressed proteins, heat maps of comparison of healthy controls with various grades of glioma using their respective differentially expressed proteins. Then the number of unique and overlapping proteins dysregulated in various grades of glioma compared to the healthy controls. MDS plot highlighting poor discrimination of healthy controls from glioma samples and using 4 different uh, dysregulated proteins. So these are the results of the different proteins that one can identify. SNX1 control grade 2. So you can see the two spots. Two spots are two same proteins printed twice to get the result for the intra-chip reproducibility, inter-chip reproducibility, along with the spot intensities and those things, and then the classifiers that whether it is showing the differential expression between control and high grade and lower grade and high grade, etc., etc. So this is how you can get. Now you can have the pathways enriched. Again, having the pathways and understanding these pathways and from where these pathways come is a different concept altogether, different software altogether, which we can show if the time permits in the context of the series of the classes and the lectures or the live sessions for that matter. We will definitely one day would go and visit that how connections or the strings or the pathways or the cytoscapes work and how we can get that. Okay. So enriched pathways emerging from the deregulated tumor-associated antigens. How the tumor-associated antigens have been found? So the proteins which spotted got into bite into the antibodies from the serum samples. We can say that this particular protein is a tumor-associated antigens, and from those dysregulated antigens, their pathways have been plotted. Their pathways have been uh, identified through different software. So what it says that the proteins attributed to these pathways, a pathway common between grades, and the blue colored grade 2, red colored proteins from grade 3, green colored from grade 4 in any pathway. Okay, so this is how you can get different pathways. And so these are the proteins, FIT signaling, PTMs, EPO, JAK-STAT pathways, etc. So the proteins which have become antigens and have developed antibodies against the normal or the known proteins of which we should not have, but due to the disease condition of glioma, it is happening, is being associated with this pathways. As simple as that. So whatever proteins that have changed due to glioma pathogenesis and have been used to generate antibodies in that particular disease person's body against the known proteins are found to be associated with this pathway. So this is as simple as that. Hopefully I'm clear and there are no doubts. If there are any doubts, please ask. Any doubts? No, right? Okay. So now we are trying to evaluate the autoantibody signatures of meningioma. Again, the same. Now, meningioma is a different another kind of primary brain tumor arising from the different locations where the gliomas or glioblastomas happens in the glial cells, whereas meningioma occurs on the meninges, right? The same experimental designs with autoantibody from patient sera probed on the chips, tumor antigens printed on chips. So that is human proteins, which is termed as tumor antigens or the tumor associated antigens, because some of them are showing the antibodies which is not normal or not should not be the case in the normal cases, which is showing differential expression with the normal protein. Some antibodies might be generated for a normal healthy persons as well, but it is showing quite different in this manner, and that's why we can say it that it is different and having the autoantibodies generated. So tumor associated antigens or tumor antigens of the human proteins which are printed on the chip, autoantibody from the patient. So patient sera will contain all the antibodies. Just flow it on experimentally, and then the finally secondary antibody, and then label channel is in that thing. Again, you can see the GST gradient. I am sure now you have understood that what is a GST gradient is that with the increasing concentration, you should have greater intensity fluorescence. That means that when we are getting particular fluorescence out in the particular having a higher expression of the intensity is not biologically relevant or not of any kind of technical interventions are there, any kind of experimental errors are there, any kind of scanning errors are there, any kind of PMT adjustment errors are there. Rather, it is a biological thing and a biological intensity which is coming different. That only we can get it. Next is the data normalization and analysis using the R, and then we are getting the control different grades of the MG1 and the MG2. So this is how it is taking molecular design and experimental design and then normalize. So these are the results again of some of the tumor associated antigens which have been showing to have, so in the C control, it is showing higher expression, and three different samples, different cohorts, different samples are to be analyzed. Again, when we are saying and analyzing using these clinical samples and the number and the statistical power for to having said that these are differentially regulated and having the different statistical considerations are need to be important. So this represents the box plots of the 15 targets from the UPROT burst onto antibody in the data, geo data set, most significant targets, and blah 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 with the different proteins, selenium binding protein. Now here again they have done Western blot as well to showcase that these particular proteins are showing a differential expression or a higher expression in the disease state, and that's why the body is reacting differently to this protein. And in the healthy conditions, it is not showing the autoantibodies, but in the disease conditions, it is showing. Again, it can be vice versa. That a control for the body has maintained a structure that to keep check particular protein to be in check, then what it is happening is that it is uh, down regulating the expression of those particular proteins. But why it is showcasing, and so it is in the uh, so why it is showcasing, but it is might be that uh, so why it is showcasing that particular thing, right? So now this is how these particular pathways, again the particular autoantigens which are with the different pathways, you can get it. Again, this is we can show you once. uh, uh, okay, I'll just, after going to it, I'll just touch upon today to make this kind of figures. Okay, to make this kind of figures, but one session we will take in a whole to have this kind of figures generated, this kind of pathway figures generated using different software. We will be using it, but for today I'll just take a small the C panels and try to find out that whether we can generate this kind of plots or not. Okay, so I am sure that it has gone. So now this another kind of arrays is the reverse phase protein arrays. After going to the reverse phase protein arrays, we will be trying to replicate this kind of figure from this kind of panel. So what is the reverse phase? So we have again discussed in detail the different types of microarrays, of which we had started with that forward phase reverse arrays, peptide arrays where peptides are there for the epitope mapping, again which we had discussed in great detail in the previous live session, where we had shown you the different workflows and different things. Now, these reverse phase protein arrays or RPPAs are also known as micro Western blotting, because it works on the principles of Western blotting. That what happens in the Western blotting, we have the cellular lysates or the proteins from the biospecimens. We take it, that protein, we do it, we run in the gel, we separate it. Just think of the principles and the application of how it is done in the Western blotting. We are running it on the gel, and after we are running it on the gel, we are getting the bands, we are transferring it, and then we are incubating with the primary antibody. If there is the protein is there, it gets bind to it, then we are doing the secondary antibody and then measuring the intensity. So similarly here, the cellular lysates are printed on the microarrays, then captured with the primary antibody. If the protein is there, the antibody which you are showing, it will get captured to it, and the quantification will be happening through the secondary antibodies. So this is the reverse phase protein arrays. So what is forward phase protein microarray and the reverse phase microarray? In the forward phase, which you have the antibodies or the proteins that you need to capture gets printed on it, and then you have the lysates, serum, anything you can say that you want to capture it. In the reverse phase protein microarray, the serum will so the cell lysates are uh, uh, getting printed on it. So this is the difference between the forward phase arrays and the reverse phase arrays. Again, please keep in mind that the micro-Western blotting is the one which is associated with the reverse phase protein arrays. Again, also keep in mind that the peptide microarrays are used for the epitope mappings, are the ones which are used for the epitope mappings. So before ending today's session, I'll just show, as I mentioned, let's uh, think of replicating this kind of figure, which we will have a good amount of session on one day for sure. We are going to one kind of software which we known as string, right? String is something that we connect, through which we connect uh, each other. So I'll just send the link here for you all to go. So I am placing on the word search, right? So now the protein name. So everyone has got this interface, right? So proteins by name, multiple proteins, protein by sequences, ranks, protein families, pathway, process, disease, organism, etc., etc. Go and click on the multiple proteins, right? Okay, so what we are getting here? Map K3. Map K3. Okay. Then what we are getting? Row A. RHOA. Then what we are getting? MYL2. MYL2. Then what we are getting? DLC1. DLC1. Then what we are getting? CDKN1B. CDKN1B. Then what we are getting? ARPC3. 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 And then what we are getting? HBG2. HBG2. Okay, organisms. Anyone? It's Homo sapiens, right? We are humans. Human proteins, human prot chips, human human protein, human proteome, right? MapK3, row A, I am guessing it's all are correct. I have no idea if it is not. Okay, all are detected. Continue. So this is a mapping step. Everyone at this step, right? Which we say that it is a mapping step. Right? Are we getting something similar to it? Let's see. Let's divide our this thing into, correct. Divide our screen. Let's see whether we are getting a similar kind of result or not. Be sure that the software has changed. Many things have updated. So let us still see. Row is in the center. Row is in the center. Row A to DLC1. Row A to DLC1. Row A to MapK3, row A to MapK3, MapK3 to the CTK19, row A is to MYL2, row A is to ARPC3 was not there and now it has been mapped. But again, we have to look at the settings. Full string network, evidence, or the confidence experiment. So if we just okay, and if we do the highest confidence, we will again discuss this in great detail in one day for sure with this thing. Here, we just looked into getting that we can replicate this kind of result or not. Highest confidence, no result, no interactions. High confidence, again, no interactions. Highest confidence with evidence, no interactions. Medium confidence, we will have the interactions. Right? So now again, if we do it, highest confidence. So any software better is to always try to use using only one particular change each parameter once and then try to find the results, right? Okay. And the medium confidence, we are getting this result. Okay. So this again I will show you. So keep in mind whatever parameters you are using, that will be helpful for you to get this kind of particular results. Okay. So this is what we can see that we can replicate to some extent with the parameters. We have definitely needs to check on the parameters and also the software gets updated, but you can see that we can make this kind of thing. We will have a complete session on this kind of pathway analysis, protein-protein interaction network analysis, of which we are talking about in the interactive sessions. Okay. So any questions? Otherwise, we can end today's class. Okay. No questions, right? Okay. Thank you all. Yeah. So we'll meet again next week. Thank you.